Okay, so good afternoon. And I'll start by thanking the organizers for inviting me. It's really enjoying the meeting and learned a lot about tissue engineering. And so I'm actually going to give a talk that's stem cell free and tissue engineering free. So I was asked to talk about the metabolism of stem cells, and our data is not quite ready. So I'm going to talk about metabolism and cell growth. So these are adult mature cells. See if I can get this to work. Here. So as we know, the heart is omnivore, and that means it can really feed on any carbon substrate. It's probably just as hungry as we are right now. We can eat anything. And, but in adult heart, most of the food the heart takes are fatty acid. And if a heart develops pathological hypertrophy and going towards heart failure, and the appetite of the heart will gradually become more towards carbohydrate, and eventually at the end stage heart failure, it also takes a significant amount of ketone. So if we're in a very exaggerated and simplistic way of thinking, the heart basically goes from blue to red with no political reference here. We all know, again, the heart has extremely high oxidative capacity. So most of those carbon that take in by the heart will get burned down to CO2. And it will generate ATP for contractile function of the heart. So for many, many years, myself included, many people here probably have considered this. What does this blue field versus red field, are they better, one versus the other, for this purpose of producing ATP to support the heart contraction? And only until very recently we realized that there's a small fraction of carbon that's not burned. It actually ends up to produce molecules for biosynthetic function and for signaling purposes. And this part becomes quite interesting to consider when we think about pathological hypertrophy of the heart as a cell growth, or maybe proliferation, maturation of stem cell-derived different cell types. So one conceptual thing here to consider is what's get burned here doesn't predict what's get used to make this. But what gets burned here might influence what gets used to make this. So for that note, I would like to share a couple of studies that were looking at what metabolism of this part and how it relates to the hypertrophic growth of the cell. So in order to study the substrate preference for adult cells, we have to first develop a cell culture model that we can feed the cells with different substrate. So Julia in the lab developed this adult rat cardiac myocyte culture that she feeds the cells with glucose, mixed fatty acid, and amino acid. And she is able to stimulate the cell hypertrophy with phenylephrine treatment. So in this model, when she stimulates for hypertrophy, and at the same time, she knocks down this enzyme called ACC2 that will promote fatty acid oxidation, and she sees the hypertrophy effect goes away. And ACC2 is an enzyme that will make endogenous inhibitor of fatty acid oxidation. Just to make sure this effect is specific for fatty acid oxidation, she used a pharmacological inhibitor for fatty acid oxidation. And when she does that, even in the presence of ACC2 knockdown, the hypertrophic effect comes back. So it is the increase in fatty acid oxidation that could antagonize the hypertrophic effect of phenylephrine. So why does that happen if we just switch the fuel from between red and blue? We will see the hypertrophy effect change. And what Julia found out is if we increase fatty acid oxidation of the myocyte, we will prevent this increase in glucose use during pathological hypertrophy. Then she take it further to show that if we pharmacologically stimulate glucose use, either use DCA or genetically stimulate glucose use by overexpressed glucose transporter group 1, we can again restore this hypertrophic effect even we knock down the ACC2. So it's shown here. The hypertrophy is suppressed, but if we override the increase in fatty acid oxidation and glucose use, and we can restore the hypertrophic effect. So this seems to suggest that the increased glucose use is required for cell growth, and that this can be antagonized by increased fatty acid oxidation. So we start to ask, what's the magic of glucose for cell growth? So Dan Shaw in the lab decided to ask this question using a mouse model that we overexpressed glucose, insulin-independent glucose transporter group 1, so that we basically dumped a lot of glucose into an adult heart. This doesn't really happen in a normal adult heart. Interestingly, in the transcriptome analysis, very consistent, quite robust change, what she found is a downregulation of branched-chain amino acid catabolism pathway. So this was an early study that we actually did microarray analysis, and when we validate with the real-time PCR, almost every enzymes in the branched-chain amino acid catabolism pathway downregulated by more than 50%. And she validated this in cell culture models that when she put the cells in either high glucose media or overexpressed glucose transporters, and she see again the downregulation of branched-chain amino acid catabolism enzymes, and in both non-cardiac and cardiac cell types, and she put them in high glucose culture, the branched-chain amino acid will accumulate in the cell. Why the whole branched-chain amino acid catabolism pathway will downregulate when we put cells in high glucose condition? It turned out that the glucose would suppress this transcription factor called KL15, and KL15 is a masked transcriptional regulator of branched-chain amino acid catabolism. You can see that here you put cells in overexpressed group 1 in the cells, KL15 level will go down. Similarly, put the cells high glucose condition, it will do that. If we overexpress KL15 and then put the cells in high glucose media, we don't see downregulation of BCAA catabolism enzyme anymore. So the KL15 is the mediator between glucose and the BCAA catabolism. So why is this important? I will skip the mechanism why glucose downregulate KL15 and just talk about why this mechanism is important for cell growth. And KL15 is a known negative regulator of cell growth. And here, if we take the neonatal cardiac myocyte, stimulate it with PE, and it will hypertrophy, and the KL15 expression will go down. But if at the same time we overexpress KL15, and the cells no longer hypertrophy. So in those cells, if we at the same time knock down the branched-chain amino acid catabolism by knock down the PP2CM, which is a phosphatase that activate BCAA catabolism pathway. If we get rid of PP2CM, and the KL15 can no longer antagonize hypertrophy. And so this observation suggests that KL15 suppress cell growth by promoting BCAA catabolism. So does this, basically here we identified a regulatory circuit, which is glucose, KL15, and BCAA catabolism. We want to know if this mechanism applies to the pathological hypertrophy of cardiac myocytes. And so we looked at a couple models. Here is the PE-induced pathological hypertrophy. We could see that when we treat the cells with PE, the KL15 level goes down. If we remove the glucose transporter, restrict the glucose uptake, the KL15 expression will come back. And similarly to KL15 levels, and the BCAA catabolism genes goes along the same pattern. In the mouse heart, if we induce a pressure overload, induce pathological hypertrophy, the glucose reliance will increase here, showing the increased glucose oxidation in these hearts. And we could see the KL15 level as well as the BCAA catabolism genes goes down. if we take a heart that we delete ACC2 in the heart, and we can maintain the fatty acid oxidation, prevent the heart goes toward glucose, and we don't see downregulation of this pathway at all, and the heart won't hypertrophy. So why the glucose KL15 BCA catabolism could change the hypertrophy response of the heart? And we are quite puzzled at the very beginning, because if we looked at heart like these hearts, we overexpress glucose transporter. It has a lot of glucose in there, and the KL15 is down, but it doesn't automatically hypertrophy. And we look at this heart that has no PP2CM and it has defective in branching amino acid catabolism, it doesn't hypertrophy either. And when we culture the cells in glucose and non-glucose media, the size looks the same. So this mechanism is not a trigger for hypertrophy. But if we're putting a trigger for hypertrophy, like stimulate the cells with phenylephrine, and then the glucose dependence shows. If there's no glucose, the cells doesn't hypertrophy as much as there is glucose. And so in those cells, if we look at the growth pathway, look at mTOR signaling, either mTOR phosphorylation itself or downstream of mTOR, and what we see is there has to be glucose. This pathway will be activated. If there's no glucose, the activation of this pathway is quite minimum. And so based on that, we developed a hypothesis, and that is like one driver and one checkpoint hypothesis. So the glucose, high increase in intracellular glucose that lead to branching amino acid accumulation is a checkpoint. It's a metabolic requirement that needs to be met before the cells decide to transmit the hypertrophic signaling for growth. And so to just demonstrate if this mechanism works in vivo, and Dan and OT teamed up to do this experiment using either wild-type mice or the defective branching amino acid catabolism mice. What they did is they generated tag to induce pathological hypertrophy. At the same time, these hearts will switch their fuel preference from fatty acid to glucose. And then in these hearts, they introduce adeno-associated virus-mediated KL15 expression to restore the BCA catabolism. So if our hypothesis is correct, in this wild-type animal, the KL15 expression will be restored and BCA catabolism will be normalized, and the mTOR activation will be suppressed, and the hypertrophy should be reduced. Which is the case as we see here. And so this is 4 weeks after a tag, and you can see the wild type with GFP treated animals has significant increase in myocyte cross-sectional area, and if we overexpress KL15, and we don't see that. And that doesn't totally prove our hypothesis yet. What's really important is in this mice that's defective in BCA catabolism, and if our hypothesis is correct, KL15 cannot rescue, because downstream of KL15, the BCA catabolism pathway is dead. And that, to our please, that it is true that we don't see the reduction in myocyte hypertrophy in the mice that has a defect in BCA catabolism. So put this together, basically this was the scheme that we were working on, and we showed that the increased reliance on glucose will allow glucose to trigger this signaling pathway that allow the mTOR be activated during the pathological stimuli for hypertrophy. What we haven't address is what about the synthetic function of substrate metabolism. So in the next couple of minutes, I will quickly address that. So we went back to the adult cardiac myocyte culture and did a proteomic analysis. Our targeted proteomic metabolomics analysis showed that in the cells that we trigger hypertrophy, we see increased metabolites from glycolysis pathway and the TCA cycle intermediate. And this is because the glucose will come down this pathway, produce intermediate, compared to the fatty acid, it will just directly go to the acetyl-CoA, go into the TCA cycle. And so we also see the pentose phosphate pathway is an increase, and that would produce more ribose 5-phosphate that is important for nucleotide synthesis. We've heard earlier this morning about stem cell proliferation. And what's interesting here is in the hypertrophic cardiac myocyte, it's not only the glycolysis pathway is increased, the TCA cycle metabolite is increased, but if we increase the contribution of fatty acid to the TCA cycle, and it suppressed everything. It not only suppressed, it not only pushed glucose out from glycolytic pathway, it also reduced the TCA cycle intermediates, and it reduced the amino acid content of the heart quite significantly. And so these are some summary data. So here I'll go quickly to two metabolites of interest, and these are the glutamine and the aspartate. Both metabolites are essential for nucleotide synthesis, and both of them are non-essential amino acid. That means they can be taken up from the culture media, or they can be made by export intermediate from the TCA cycle. And what's really interesting here is very different from proliferating cells that we've heard a lot about glutamine addiction of cancer cells or proliferating cells. And there is no glutamine addiction in adult cardiac myocytes when you stimulate for hypertrophy. So here we stimulate for hypertrophy. The glutamine level in the hypertrophy cells are normal, and they don't really increase glutamine uptake from the media either. But interestingly, if we push fatty acid oxidation, we could suppress the glutamine consumption from the media, and the glutamine content of these cells are much lower. It's a different situation for aspartate. Aspartate level is very high when the cells are stimulated for hypertrophy. Aspartate is critical for nucleotide synthesis, you'll see later in the next slides. But this aspartate is not coming from media. The uptake of aspartate from media is not different at all. Doesn't matter we stimulate hypertrophy or we push fatty acid into the cell for oxidation. The cell do not take more or less aspartate from media. And so we ask the question, where did the cells get these things, and why it has less of those? And we suspect it's because the difference in glucose use. And so to look into that, we labeled glucose with C13 carbon, and we tracked where the glucose go in these cells. So if the glucose come down this way and join the TCA cycle, then the intermediate of TCA cycle metabolite will have 2 carbons, that is C13 instead of C12 that would come from other substrate. So if we do a mass spec tracking, we could see clearly that when we stimulate hypertrophy, we got more contribution from glucose for those metabolites such as citrate, alpha-ketoglutarate, and glutamine, and aspartate. But if we push in more fatty acid into the TCA cycle, fatty acid got into the TCA cycle, it could be burned down by CO2, but it will compete glucose out of this TCA cycle, and they will suppress those metabolites. And that will lead to decreased aspartate level in the cell. So as I mentioned, ribose 5-phosphate, aspartate, and glutamine are all important for nucleotide synthesis. Even pathologically hypertrophied cardiac myocytes do not proliferate. They don't need to synthesize the DNA, but they do need to synthesize RNA to increase transcription and for protein synthesis. So we showed that if we increase fatty acid oxidation, we prevent the glucose getting into the cell, we could suppress this increase in pure nucleotide in hypertrophied cells. To demonstrate this is really a causal relationship, we cultured cells that we deliberately increased aspartate supply, either by cell-permeable aspartate or just dumping huge amount of aspartate in social media. And we can restore this hypertrophy response even in the presence of ACC2 knockdown. And similar thing can happen if we just supply a lot of nucleotide. So this is just to show this, we find a similar mechanism in vivo that the ACC2 knockout mice that has high fatty acid oxidation has decreased hypertrophy. It has lower ribose phosphorus level, and also the aspartate level is no longer increased. So to summarize it up, we found several mechanisms that the increased glucose reliance contributes to the pathological hypertrophy. One mechanism is the signaling pathway that would facilitate mTOR signaling and then stimulate protein synthesis. The other pathway is through either the pentose phosphate pathway or TCA cycle that will contribute to nucleotide synthesis and the gene transcription. Both of them could contribute hypertrophic growth. And what's interesting for the substrate selection theme is that if we promote fatty acid oxidation and prevent the increase in glucose use, we can actually suppress those mechanisms and reduce pathological hypertrophy. So I want to thank the team, and we're really a fun, vibrant group of people, very talented, and I've already mentioned Dan and Julia and Otis' work, and other people working on other projects. And our funding agencies, critical collaborators, and Rick helped me to make the Group 1 transgenic mice, and Dave Olson provided ACC2 flox mice. The BCA catabolism defective mice are from EBIN1, and Roger helped the AAV9 vector, and Dan Raftery helped with metabolomics analysis. And thank you very much. Yes.
In the mouse where you inhibited the BCA catabolism and you prevented the hypertrophy, what was the phenotype from a functional contractile point of view?
So the BCA catabolism defective mice has no baseline phenotype, but they do very poorly after pressure overload. So EBIN1's lab reported that in 2 or 3 years ago, and we reported that they do poorly after ischemia reperfusion as well.
Do you have any insights if whether any of these would change in, during exercise or in physiological hypertrophy?
Yeah, that's a good point. And I think we did, for the glucose BCA relationship, it looks like it applies to physiological hypertrophy as well. And it's not a complete study we did.
Okay.
Thank you, Ron. I think in the interest of time, we'll conclude this session now. Please join me in thanking all of our speakers for an excellent session. We'll break for lunch now. We're back in here at 20 past.